Department Head of Entomology at Penn State University. Dr. Falton attended the University of California, Irvine for his undergraduate degree. He then earned his master's degree at Kentucky University and his doctorate at the University of California, Davis. He was employed by the University of Arkansas for 10 years, 10 days, and 22 minutes. But who's counting? Not Dr. Falton. He then moved on to work for Penn State for 19 years and counting. His research focuses on the chemical ecology of plant-herbivore interactions. And everyone please join me in welcoming Dr. Gary Falton.
Well, thank you very much. Actually, as a professor, I think one of the biggest honors is to be invited by a grad student group, and I consider that a honor. In today's talk, I'm going to talk about work that's primarily from one of my own grad students, just really one who did almost all of the work. So this is a very student-centered talk. The system I'm going to talk about actually goes all the way back to my master's project. So I've never given up the group of insects I've worked on, so I'm kind of come full circle on this. So let's get started. We all have parasites. How many of you think you have a parasite? Pretty good. Most of you do. It'd be surprising if you didn't. You know what the most common parasite in people generally is?
Teenagers?
What? Mites. Yes, hair follicle mites in your eyelashes and eyebrows. A large percentage of the population has those. There's other parasites that a lot of people have across the world. The protozoan toxoplasma is very common in some countries. 80% of the people have this parasite. So parasites are everywhere. And what I think is kind of cool about them is that in some cases, they really generate very interesting behaviors and phenomena in your host. And some of these are the most sort of bizarre stories that you can imagine. And I think the interesting thing about it is not just the fact that they're interesting behaviors, but today, with modern tools, we can actually look at some of the mechanisms that help explain those behaviors. So that's sort of what I'm going to talk about today. What I want to do first is to give you just a feel for some of these sort of bizarre behaviors, and then we'll talk about our own work. Does anybody know why lemmings jump off cliffs into the ocean? Any ideas? It's actually not true. It all goes back to a Disney movie in the 1950s where they actually forced these animals off the cliff. They do mass migrations, and they may end up in the water and other things, but they don't commit suicide. So this is not a true phenomenon. But there actually is one among insects, and this is a very interesting one, the crickets. They actually do commit suicide. And why do they do that?
Yes. Oh, I'm sorry, I should have answered the question.
You got the answer. This is so common that in some streams and ecological niches, that most of the food fish may eat are actually crickets and other insects that have committed suicide. So let me show you a video of this. So this cricket is going to jump into the water. And it actually, I guess it doesn't know how to swim because it's going to die. And about the time it dies, a worm starts coming out of its body. And that's a long worm for that insect. So what they do is they manipulate the behavior of crickets, other insects, that normally are not aquatic to go into the water. So why do they do this? Well, it's because of their life cycle. The worm actually becomes a free-living worm in the water, It lays eggs, forms larvae, and those end up being re-ingested by other insects, such as mosquitoes, that could then come out of the water as adults, and then other insects eat those, and it completes the life cycle. But as I said, in some areas, this is so common that most of the diet of certain fish may be these dead crickets or dead praying mantis or whatever. The focus of our work is with parasitic wasps. And these are an interesting group of insects. There's hundreds of thousands of species of parasitic wasps or parasitoids. So it's a very common group of insects. And they're known to manipulate a lot of aspects of their host in order to survive. Typically, these parasitoids inject eggs most often inside the host. The egg hatches into a larva, it feeds off of the blood of the host, or sometimes it's internal organs as well. So I'm going to give you an example of several of these. This happens to be one of them that I work on. And that parasitoid is sort of tasting the host to make sure it's the right one, and then it injects an egg and it injects venom into that host. This is what happens when these parasitoids develop. They develop into larvae, and when the larvae are done feeding, they come out of the host. And this is a tobacco hornworm that has a parasitoid that lays hundreds of eggs inside that host, and eventually they come out. They'll pupate and then form a wasp that will go in search of another host. Those are the cocoons for the pupal stage of that wasp. And you can see one emerging now from the host. This is a really bizarre parasitoid. And it only parasitizes cockroaches. And what it does is it injects venom into the brain of the cockroach, and it's able to find a very specific part of the brain of that cockroach, and it causes a very weird behavior in the cockroach. I'll show that to you here. That little wasp is leading the cockroach around. It's going to take it to a secluded spot so that its egg and larva can develop. You know, obviously the cockroach is a lot bigger if it can redirect the wasp could, but its brain has been affected by this venom that was injected.
Wow.
Just pretty bizarre behavior. Here's another one that parasitizes spiders. And there's a whole group of these parasitoids that parasitize large number of spiders. And they manipulate the host, again for their own benefit, and they do it in a very different way. This parasitoid typically lives on the outside of the spider, develops on the outside of the spider while feeding on the internal blood and organs. And these are the webs of a group of spiders, the way they're supposed to look. What happens when these spiders are parasitized is their webs become very different. And their webs are basically protection for the wasp pupal stage. So they form a web around that that protects the parasitoid from predators. So it changes the whole spinning behavior of that spider. That's a close-up of it. You can see cocoon of the parasitoid in the middle of the web on the right. Obviously, a very different spinning pattern. It's just totally changing the behavior of that spider. This is another interesting one related to caterpillars. And this parasitoid causes a different type of behavior. And what happens is this parasitoid lays a number of eggs inside the caterpillar. These eggs develop, and some of them, the larvae come out, form cocoons, as you can see on the stem of the plant. But a few of these linger behind in the host. But they cause that caterpillar to do something very interesting that normally they would not do. First of all, the caterpillar stays right near the cocoons. It doesn't leave the site or move about. It stays in the general area. And if there are any predators in the area, what happens, look, I'll show you with this video. There's a predator coming up. So they go through this body guarding behavior and knock the predator off. And this has been measured with actual data, and you can see the parasitized caterpillars are much more likely to exhibit this behavior. And they've shown that it actually does increase the survival of the parasitoids because it protects it from potential predators that may eat them. Another related example is with the ladybird beetle and a wasp that parasitizes that. And it's sort of mentioned that they turn the ladybird beetles into zombies or whatever. And what they do is they do a body guarding behavior the same way. Basically, after the larva emerges from the host, the ladybird beetle then sits on top of the cocoon and presumably protects it from predators attacking. So they stay with the parasitoid, they stop looking for food for themselves, they eventually die. But that's sort of a body guarding phenomenon that's similar to the other example. What they found in this system is most of these examples I've shown you, they don't really understand the underlying mechanisms. They may know it's a venom, they don't really know what's involved with this. But in this example, this behavior is due to a virus that's associated with the parasite. So the virus itself is mediating the effect, and not the parasite. Okay? Interesting phenomenon. The system I work with, and I started this work with this parasitoid and caterpillar when I was a master's student. We didn't work on it until about 5 or 6 years ago. We came back to it, a student of mine wanted to work on this project, and I think developed some really interesting hypotheses. We wanted to see how this parasitoid may impact the host insect and improve its performance in ways that people hadn't looked at before. So in this example, this parasitoid obviously is stinging the host, injecting venom. What happens is they feed internally on the host. They feed on just the blood. When they're complete development as a larva, they then emerge from the host. You can see the picture on the top right. It's almost as big as a caterpillar. It's pretty large, and all it's been doing is feeding on blood for the last 7 to 8 days. This is an interesting system because there's a couple of families of parasitoid wasps, Riconidae and Nomadidae, if you're interested in taxonomy. But they carry a virus. It's called a polydNA virus. And many years ago, several decades ago, this virus was identified, and it was the first example of a mutualistic virus. So it actually benefits the parasitoid. And how it benefits the parasitoid is a very interesting phenomenon. So it said besides the egg and the venom, they're injecting the virus goes right into the caterpillar along with the egg. And several decades ago, in classic work done by a couple of entomologists, and these decades being one, is that they found that this virus suppresses the immune system of the caterpillar. So it's benefiting the parasitoid in a very specific way, because if the virus is not present, they did these experiments, which are really tough to do, is really rinse off the virus off of the egg and then re-inject it into the caterpillar. And what happens if the virus isn't present, The immune system is upregulated and it attacks that larva and kills it, so it never develops. But in this example with the virus, it is able to suppress the immune system. There's been quite a few studies done since then by other labs where they've identified all the factors involved with immune suppression. It's been brilliant clinical work. These viruses only contain about 100 genes, so they're very small genomes, but those 100 genes may do a lot of different things to the host. The interesting thing about this virus is it only replicates in the ovaries of the female wasp, in a specific region called the calyx. If you see some light blue area, that's actually fluorescence from the virus. There's a lot of it in this part of the ovary. It bathes the eggs as they're ejected into the host. And the virus, as I said, only replicates in the wasp. What it does in the caterpillar is interesting. There are maybe a subset of those 100 or so genes that are expressed in the caterpillar, and they specifically stop the immune system from functioning. But the virus itself doesn't replicate in the caterpillar, so it's an end host, but the parasitoid obviously keeps it going through the generations. So the question my student had was, we weren't looking at behavior in the system, we were looking at feeding of the caterpillar and eventually impacts on the parasitoid. We wanted to know if the parasitoids altered caterpillar feeding to the benefit of the parasitoid. So is there some aspect of their feeding behavior that changes when they're parasitized? What we found was, I think, surprising finding. Before I tell you what that finding is, I need to tell you a little bit about induced defenses in plants, and specifically our system that we'll talk about today is in tomatoes. When plants are wounded by chewing insects, they elicit a wide range of different biochemical reactions, one of which is regulated by the plant hormone called jasmonic acid. And jasmonic acid causes upregulation of certain plant defenses. And in tomato, there's a whole suite of defenses that I call anti-nutritive proteins. And these include the ones I'll talk about today, polyphenol oxidase and protease inhibitors. There's a whole bunch of different protease inhibitors in tomato, but as a suite, they interfere with the insect's, the caterpillar's ability to acquire nutrients, either by inhibiting their digestive processes or actually binding to the nutrients and preventing them from being used by the insects. So these two defenses are the ones I'm going to keep referring to. So what happens when a parasitized caterpillar feeds on tomato versus a non-parasitized? This is a measurement of polyphenol oxidase activity. There's a background level when the plant's unwounded. When it's fed on, it goes up. And in the case of the parasitized caterpillars, it didn't go up nearly as much. And what I need to tell you about this experiment was that the amount of feeding was identical. We restricted the insects to a caged area, so both the parasitized and non-parasitized caterpillars fed the same. So the amount of damage is the same. So why is there a difference in this plant defense? The interesting thing about polyphenol oxidase, to bring home the information about it to you, it's involved with the browning reaction that you see in apples, you see it in bananas. It's oxidation of phenolics, which interferes with the use of nutrients. So you see this all the time with fresh fruit, fresh vegetables and such. The browning reactions due to this enzyme. So why would we see a difference in the plant defenses between these two different treatments, even though the caterpillars ate the same? What these caterpillars do when they feed is they're actually salivating an awful lot. They're dumping micrograms of protein and other chemicals onto the plant as they feed. So they're actually very messy feeders. They dump drool saliva all over as they're feeding. And there's a lot of functions of that saliva. I'm not going to go into all those today. But the saliva originates from primarily from a pair of what we call labial glands. Those labial glands converge into a structure called the spinneret, which is actually underneath the sort of the jaw of the insect. So it's actually outside of the digestive system. So as the insect's feeding, it's just shooting out saliva all over the place. So we've done a lot of work on saliva over the years. This is some of the earliest work on proteomics of the saliva, wanting to know what's actually in the saliva that might be having these effects that the insect feeds. And when we first did this work, we found that a large amount of the ejected or secreted protein was this enzyme called glucosoxidase. And Richard Musser's paper in 2002 sort of made that famous. But glucosoxidase is an interesting enzyme. It oxidizes glucose in the presence of oxygen and produces hydrogen peroxide and glutamic acid. Hydrogen peroxide has a whole bunch of different functions in plants, also in insects. And one of the functions of this enzyme is because hydrogen peroxide is antimicrobial, what they're doing is they're, in a sense, sterilizing the leaf as they're feeding in case there's any pathogens on that leaf. So they're inhibiting bacteria and other microbes on the leaf. That's one of the functions of glucosoxidase. But one thing it does do to tomato plants is that it actually induces these defenses. Protease inhibitors, as I mentioned. If you take saliva directly from the caterpillar, apply it to the plant, apply it to a wound, you get an elevated response. You apply purified glucosoxidase, you get the same effect. So this is part of the reason why you might see different responses in the parasitized caterpillars. It could be due to the saliva that's being secreted. So our first question obviously was, does a parasitoid regulate the glucosoxidase in the caterpillar? And yes, it does. After about between 3 and 4 days, we begin to see a suppression of the gene expression for this particular enzyme. We also see a suppression in the enzymatic activity. So the parasitoid is down-regulating the salivary enzyme. And it obviously has consequences. So our question at that point was, well, how's a parasitoid regulated? And we had several different hypotheses. One of which is these parasitoids, as they're feeding inside the host, are taking up a whole lot of resources from the host. So it's possible it's just a depletion of resources in the host, and the host can no longer make a lot of this glucosoxidase. That's certainly possible. It's really, really hard to determine that. It's not an easy experiment. The other aspect could be an injection of venom. We can collect venom from these venom glands and inject it into the host and see if that had an effect on glucosoxidase. By the way, it didn't. So it goes back to this virus. Is the virus mediating the suppression of the glucosoxidase? So what we found was that you can actually purify this virus. It takes a lot of dissections of these ovaries of these parasitoids, which are very, very tiny. The good news is that they're so filled with a lot of this virus that through differential centrifugation, you can actually get a purified virus that we could then re-inject into the host to see if we would get the same types of responses. And as you might expect, that's what we saw. So we took 1/10 of the female equivalent of the virus, whether or not that's how much the parasitoid injects, I don't know, but it's a starting point. So what we saw was we saw the same level of suppression, basically, of glucosoxidase as we did with the parasitized caterpillar. So it looks like the virus is affecting the salivary glucosoxidase. And remember, there's less than 100 genes of this virus that's